on the exam. I took the kidneys off, but just want to review some of the material here about the kidneys before I go on, because I didn't finish it. First of all, the anatomy. Okay, so the anatomy of a neuron, or not a neuron, I'm already up at that point. Nephron, okay? Nephron, okay, first of all, Bowman's capsule, okay, that's the important part, connected to proximal tubule. It's called proximal convoluted tubule because it's really twisted all around, so that it has a lot of surface area, okay? That runs into the descending loop of Henle, then the ascending loop of Henle, and this is on a juxtamedullary nephron, okay? Cortical nephrons do not have a long loop of Henle. Now this is very significant with these juxtamedullary nephrons because they penetrate deep into the medulla, okay? And that's real important because in the medulla, going from the cortex into the medulla, there is a concentration gradient, solute concentration gradient, and that is very important for bringing back water into the, absorbing water into the bloodstream. Then this runs into the distal, convoluted distal tubule, which then runs into collecting ducts, let's say. And this collecting duct then brings the filtrate, by this time urine, down to the renal pelvis, through the ureter, down to the bladder, okay, single bladder. And when that stretches enough, then you are given a reflex wanting you to pee, okay, and then the rest of it is the bladder will empty through the urethra. Okay, so that is the nephron and the collecting ducts. The collecting duct, it does not take just only the filtrate from a single nephron, but from several neighboring nephrons, okay, including cortical nephrons. Blood supply, which is being filtered. The input is the renal artery from the aorta. So go through the aorta, renal artery into an afferent arteriole that goes into the Bowman's capsule, breaks up into a capillary bed, glomerulus, and this is where fluid comes out, okay? So it's a common theme that we've had before of epithelial, of material leaving at a capillary. Blood pressure is very important here to push material into Bowman's capsule, out of the bloodstream. And so this is ultrafiltration, so it's basically plasma, or serum that comes in there, because large blood proteins and blood cells, of course, stay in the blood vessel. This filtrate, okay, continue with the bloodstream. Then what leaves glomerulus is an efferent arteriole, which breaks up into a capillary bed that surrounds both the proximal and distal tubule. And this is called the peritubular capillaries. Then that blood will move into another capillary bed called the vasa recta. However, there's a descending vasa recta and an ascending vasa recta. The way the blood is directed is that the blood is going descending in the part that surrounds the ascending loop of Henle. Okay, this is countercurrent. On the other side of the vasa recta, the blood is moving upward, and it is surrounding the descending loop of Henle. Once again, countercurrent. There's a reason for that, okay, or a function. And also this, even though this is drawn with the collecting duct further away, it's serviced also by the vasa recta or other types of capillaries that we don't need to worry about. All right, so what goes into your nephrons collectively is about 180 liters a day. And so that's a lot of water. And most of that water needs to be brought back into the bloodstream. Okay, typically you only lose about 1.5 liters a day in the urine. So that's real important here as to what's going on. All right, so I got into this, filtration, secretion, and ultrafiltration. Okay, that happens, of course, in the glomerulus. And then filtration, secretion, and reabsorbance happens all along. Okay, but it depends on where, what is being brought back in. And so at the proximal tubule, as I already discussed, primarily driven by the sodium potassium ATPase. Okay, salt gradient is set up so that many of the organic material, like glucose, other monosaccharides, and amino acids, are all reabsorbed here. Okay, and we already went over that, and so I'm not going to go into details here, right? And so where does that material go? It crosses the epithelium into the interstitial fluid, then crosses the endothelium of the peritubular capillary. So this is one common theme, two epithelia being crossed. Two epithelia being crossed. And water follows osmotically. So we reabsorb some of the water here. Then when it goes down the loop of Henle, what we have on the interior, going down the medulla, is a salt gradient. And it's mostly made up of sodium and chloride. Okay, sodium chloride ions, and it gets more and more concentrated the deeper into the medulla you go. Okay, now one of the important things here about the descending loop of Henle is that it is impermeable to salt, but permeable to water. How can it be permeable to water? Because the plasma membranes of the epithelium have a large number of aquaporins. Other than that, it has watertight plasma membranes, and the only way that the water can come out is through the aquaporins. And so, first of all, it starts out isosmotic with the body. Okay, body fluids is about 300 milliosmoles. And as this material now moves down into this gradient that's on the interstitial fluids, first of all, we have 300, now it's 500 outside. Water will move out. And as water moves out, the salts become more concentrated, and therefore until they reach equilibrium. Okay, because the salts can't move. Salts have to stay. It's impermeable to sodium chloride. So this salt gets concentrated until it's at the same level as outside. Now what's happening to this water? It's going into the ascending vasa recta. Okay, so it's coming out of the loop of Henle into the vasa recta. Those capillaries are right next to the descending loop of Henle. And then as that moves down, it becomes more and more concentrated as we take water out. And therefore it can become, in humans, as concentrated as 1,200 milliosmoles. Now there are some rats that live in the desert, kangaroo rats, that they can concentrate their urine up to about 2,200 milliosmoles. It really, really concentrated. And that is because with regard to the size of with respect to the size of their kidneys, they have real long loops of Henle. Okay, that allows them to get even more concentrated. Why is that important for a kangaroo rat? They eat seeds, they live in the desert. They never drink. They never drink. So where do they get their water?
Their fruit?
What?
They're like fruit?
Seeds don't have a whole lot of water in it. Water preservation is obviously very important to them. That's one thing. The long loop of Henle allows them to really concentrate the urine so that they are not losing a whole lot of water. Furthermore, behaviorally, they're not out during the day. They're usually down in burrows during the day so that they're not subject to a lot of evaporation. But they never drink. They still lose water. And that water has to be replaced. So where do they get the water?
From underground.
What?
From underground?
Nope, they don't drink.
What?
They don't drink. So if they're underground, how are they going to get water?
They don't get their water?
Huh?
Like they don't drink water then?
They don't drink water.
They absorb it through their skin.
That doesn't happen. Think of Biology 101A. What was one of the major metabolic pathways we went through for Biology 101A? Besides photosynthesis. Obviously animals don't do that.
Cellular respiration?
Cellular respiration. What's the summary formula for cellular respiration?
Oh.
They make water.
Yeah.
They like inhale.
Water is 12 moles. Okay, you have 6 moles on the reactant side of water, 12 moles on the other. There's a net of gain of 6 moles of water, metabolic water, just from the breakdown of the food. Metabolic water, that's all that they live on, okay? Just their metabolic water. And this here allows them to really concentrate their urine. Okay, then as we go around the loop of Henle, and going back up, now we have concentrate, here the membranes of these cells change. They have no aquaporins. Water cannot exit, but they have channels for sodium chloride. Okay, they have channels for sodium chloride. So as this concentrated filtrate comes up into an area where in the interstitial fluid you have lower concentration than what's on the inside, and water can't move. Water can't move. It is the sodium chloride that's coming out the channels, down their concentration gradient. They have a higher concentration gradient inside than outside, and therefore this would diffuse out until equilibrium. Once you get up to about 700, then their sodium potassium ATPase actively transport sodium out. All right? And this is part of what adds to and creates this gradient. It isn't the only thing. Yes?
So what's the name of this rat?
Kangaroo rat. Okay? And there's others. There's other rats there too. All right? And so as this is getting, and so because of active transport, by the time we get up here into the proximal or distal tubule, we can have the filtrate hyposmotic, okay, down to as low as 150, and then as because of body fluids, 300, okay, and it's here where we have another area of secretion and reabsorption of various types of solutes. Now it puts up here bicarbonate, and of course hydrogen ion can be put in. Sodium chloride, there's a lot of sodium chloride transporters up here. And once again, that's active transport being transported out. But this is also where other minerals such as phosphorus, magnesium, calcium is adjusted as well. Okay, it's adjusted. Now, if you have plenty of water in your body, in fact, too much water in your body, as we go down the collecting duct, what happens in the collecting duct is it is variable. It can change, alter its water absorption properties. And so if you have, you've been drinking a lot of water, and therefore you need to get rid of a lot of water because you have excess water in your body, these membranes are modified so that no aquaporins are there. The aquaporins are removed, and therefore water cannot exit. And so even though this 150 millimolar concentration is coming deeper and deeper, even if it is 900 on the outside, there's no way for water to exit. No way for water to exit. And if water can't exit, then what you urinate is hyposmotic. All right? If on the other hand, you have you've lost a lot of water, then aquaporins are inserted. Aquaporins are inserted that allows water to exit. And of course, where does the water go? It goes into the vasa recta. Furthermore, down in the distal part of this collecting duct happens to be, it's permeable to urea. Now, what's been happening with urea along here? First of all, it was very dilute up here. But as we remove more and more water, as more and more water is taken out, by the time we get down here, urea is pretty concentrated. And it can be allowed to leak out. So that contributes to this gradient in the medulla. So the gradient is set up by active transport in descending loop of Henle, as well as leakage of urea down here, and that could be moderated. Now what happens to this urea, it gets into the interstitial fluid, and then it just simply goes back into the ascending loop of Henle, and goes back around. Okay, so this is how the system is working. Let's go to, let me see here. Yeah, so what sets this up? It's called the fact that the two ascending loops of Henle are going countercurrent to each other. This is not called countercurrent exchange, however. It's not countercurrent exchange, but the fact that those are running countercurrent to each other, it is a countercurrent multiplier. It's what causes the gradient. So let's look at that countercurrent multiplier here for a moment. Okay, this is what I made. Give you some idea. If we started out in equilibrium in all three of the compartments I'm talking about, here within the ascending, descending loop of Henle, interstitial fluids, and ascending loops of Henle. Okay, this is what we have. What is driving this, of course, over here, sodium chloride transporter. Sodium, potassium, HPAs, pumping sodium out into the interstitial fluids, and chlorines following the charge. So if we're pumping that out, sodium chloride out here, and water can't move, what's going to happen to the concentration here? We're pumping salt out.
It's gonna decrease.
It's going to what?
Decrease.
It's going to decrease. All right, and it's going to decrease there. Whoops, went the wrong way. Sorry about that.
What is that?
Just a second. Somehow I jumped. Yeah. Okay, so, and furthermore, this concentration here is going to go up, right?
So why didn't the right side decrease on this one?
What's that?
Why doesn't it show the right side decreasing on this slide?
Why what?
Why didn't it show the right side decreasing on the next slide?
Well, because I hit the arrow going the wrong direction. Start at the very beginning. Okay, so this is the first part, that's coming out. I'm doing it step by step. Okay, so as this is going out, sodium chloride, what happens is, first of all, that increases, but the inside decreases too. Okay, so this is becoming less concentrated, this is becoming more concentrated. However, this is moving, right? Okay, so if we move this down just a little bit, oh, and also what's happening here too, is on the other side, water is coming out. And so what's going to happen to the concentration there? It's going to increase till it's in equilibrium. All right, so we're moving the water. So now we have fresh 300 millimolar that comes in. All this stuff is moved up here and into the proximal tubule, but we're still having here sodium chloride being pumped out. And so what's happening here then is, of course, this becomes concentrated up here, but because this was 200, that becomes maybe 350, but because that down here was 400, that becomes 500, and so forth, okay? Because the concentrations now are not equal along the side here. And inside, this is becoming more dilute. But down here, since it started out as 400 to begin with, or it lags behind what's happening up here. And we do it again. Moving out the water, and then so forth. Okay, so what am I doing here? Yeah, keep doing this. So what you should see is, because of these two loops of Henle going counter to each other, and the flow coming in counter current, we're starting to set up a gradient that increases as we go into the medulla, decreases as we go from the medulla to the cortex. And this is what sets up the salt gradient. In addition, see, I think that's all. What is this? Oh yeah, this is when I animated so that it moves. Okay, I can't complete that, it was too complicated to do.
Pretty good.
Huh?
Pretty good. It's pretty good.
Yeah, but it took a lot of work to get there, and I just said, I'll
We appreciate the effort.
what's that?
Said we appreciate the effort.
Okay, I'll see how far I got with it. Okay, we've already gone through all this. Okay, there, everything's in equilibrium. Moving out the salt, changing the concentrations, water moves out, and that moves. Okay, then I go back to this, and so forth. Anyway, should give you some idea. Okay, now, another important thing here is, okay, so that countercurrent multiplier is talking about countercurrent flow of the two loops of Henle and the active transport of the salt in the ascending loop of Henle. However, if the blood flow were co-current with the vasa recta compared to the flow in the urine, or in the loops of Henle, the blood would eventually carry away that current, that gradient. that gradient wouldn't be able to exist. The only way we can maintain that gradient is that the fact that we have counter current flow between the descending loop of Henle and the ascending, or descending vasa recta, ascending loop of Henle. If they were going co-current, where this was going alongside here, this gradient would be stripped away. We wouldn't have a deep salt gradient. So here, because we have, first of all, blood that is 300 milliosmoles that is going in here, and we're pumping out sodium chloride here, which goes right into the ascending vasa recta. What's that going to do to the concentration of blood in the vasa recta? Sodium is being actively pumped out here, and it moves over into the bloodstream. What's that going to do to the concentration in the blood? Right here. Further,
It's gonna increase.
what's that?
Increase.
It's going to increase. Yeah, so this is going to increase because this is going to get more concentrated. Water is moving over there, even with the blood, okay? And so this is becoming more and more, the blood is becoming more and more concentrated because the salt is being moved out of the ascending vasa recta, and it goes right into the blood, which leads to its concentration. When we turn the other corner here, it's now countercurrent to the descending loop of Henle. And so as we have concentrated, and this is permeable to water. So as we're going up here, water is coming from the descending loop of Henle into the blood as the blood is going countercurrent. So here's where we have countercurrent exchange. There's countercurrent exchange of salts and waters such that by the time our blood gets up here, it has been diluted by the water that's leaving the descending loop of Henle, and it is at now isotonic isosmotic with the blood. So the blood becomes more concentrated coming down because salt moves over, and then as it moves up, it becomes less concentrated because of taking up the water, okay? And since it's countercurrent, that gradient stays there. We don't destroy it. Otherwise, if it's co-current, it wouldn't work. Okay, so furthermore, down here, this doesn't show it. Yeah, right here, some leakage of urea can come out, and it contributes to this gradient as well, okay? All right, so that's how the kidneys are working, and one of the important things is that they are under hormonal influence to balance their reactions. So give some ideas here about regulation of kidney function. So our urine can be as dilute. I said 150, that was on that figure, that actually our kidneys can make it as dilute as 70 milliosmoles, or as concentrated as 1,200 milliosmoles. That's quite a range, okay? Now, when would it be dilute?
It's because a lot of water.
When you take in more water than what you need, yes. When you drink like a fish, all right? So, and when is it concentrated?
No water.
When you're sweat and you've lost a lot of water and cannot drink to replace it. So we have this amazing range here, whereas with frogs, snakes, and lizards, they at most can get their urine isosmotic. They can make it hypoosmotic, but they can't make it hyperosmotic to their body fluids. All right, so how is this done? Several different hormone systems here, and I'm not going to discuss them all, but some of them here. One of them is called antidiuretic hormone, or ADH. It's also in some books called vasopressin, okay? It has a control here. So this is released from a gland, which I'll talk a lot about in the next lecture, the pituitary gland. In fact, there's two different pituitary glands, although they've grown together in mammals. There's an anterior pituitary, posterior pituitary. And this is just right under the hypothalamus of the brain. So close to the brainstem. So it's from the posterior pituitary that it releases antidiuretic hormone. Now this antidiuretic hormone, however, is only stored in the posterior pituitary. It is not made there. This is really connected to the hypothalamus. So we have the hypothalamus, we have up here hypothalamus, that part of the brain, and then the pituitary down here. One of the things that we have is that there are special types of neurons. They're called neural secretory cells. They're like neurons. Have the cell body up here and long axon, and then branched where it has axon terminal. It's actually these cells that are making ADH, transport it down to the axon termini down here at the pituitary, store it there, okay? Where it is later released. Now, what is the stimulus for this release? Is that there's sensory cells here as well that have stretch receptors. And so as we have a capillary comes in, and if we have high solute concentration in that capillary, where is water going to move with respect to the interstitial fluids?
Into the capillary.
It's going to move into the capillary. And so that drags water out of those cells. Causing, no, they don't collapse completely, but we're dehydrating those cells a little bit, and they have stretch receptors in the plasma membrane that they can detect that change in water. Okay, that is the stimulus that leads this axon termini to release antidiuretic hormone, which goes right into the bloodstream and carried all throughout your body. Okay, now one of the things with hormones, however, that antidiuretic hormone will only influence target cells that have a receptor for it. Okay, so target cells they have to have the appropriate receptor. And this is a receptor that is a G protein linked receptor, and it's only found in cells in the collecting duct. Okay, so nothing in the body can respond to it except for those particular cells. So this is responding to a change in osmolarity, okay, or a decrease in water content. Okay, so what type of conditions do we need our kidney to do if we have lost water?
Conserve water.
We want to conserve water, okay? So the collecting duct needs to be adjusted so that as filtrate goes down from the cortex into the medulla, that water is reabsorbed, okay? All right, that has to be, so it's target is the collecting ducts, G protein linked receptor, which you should know about from last semester, but I'll review it. And what this leads to is a signal transduction pathway through the membrane, phosphorylation cascade that activates eventually vesicles, transport vesicles that contain in the membrane aquaporins. These vessels then fuse with the plasma membrane of collecting duct cells, providing the aquaporins for water to be reabsorbed. Okay? And so this is due to an increase in the permeability of the water, and so more water is retained. So here's from your textbook. This is the lumen, and what we have, the aquaporins on the basal membrane, they are there. Okay, this is interstitial fluid out here. We have aquaporins there. Where we initially don't have aquaporins is in the apical membrane. Okay, but either way, if we don't have through either basal or apical, if there were no aquaporins, water can't exit the lumen. Okay, so we're wanting to reabsorb it. So there is ADH binds to the ADH receptor. There's G protein that stimulates adenylate cyclase, same thing that I've talked about before, adenylate cyclase, which converts ATP to cyclic AMP. Cyclic AMP binds to cyclic AMP dependent protein kinase. And what do kinases do? They phosphorylate other targets. And usually those other targets happen to be kinases. And so the attachment of phosphate leads to a conformational change, activates the kinase. So we get that's signal transduction pathway, phosphorylation cascade, that certain target that then binds to these aquaporins that are tethered, or these transport vesicles that are tethered just below the plasma membrane that contain aquaporins. And these fuse, okay? And this now allows water to move in, out, because of the gradient. And so we reabsorb water. We're moving water out of the lumen. The lumen becomes the filtrate. The urine becomes more concentrated, And this is dynamic, because once the level of this is reduced, as things come back into more equilibrium, this aquaporins are brought together into a cluster on the plasma membrane. We get invagination, so that they're removed. Okay? So this membrane is constantly being remodeled. Someone from New York, no, Walnut Creek, I don't know anyone in Walnut Creek, okay? All right, so that's one of the controls here. Now there's another hormone system that's a little bit more complicated. It's called the renin, angiotensin, aldosterone system. That's what that stands for, renin, angiotensin, aldosterone system. And this responds to a drop in blood pressure, okay? Now why would the blood pressure drop? Under what conditions would the blood pressure drop? In your body. Because the blood pressure is real important for filtration of the glomerulus. That's real important, okay? But when would it drop?
Like if you haven't eaten in a while.
When you what?
If you haven't eaten in a while, don't you get like?
Okay, what happens if you're sweating a whole lot? You're losing water. Do you think your blood pressure would drop if you lose fluids, or if you cut yourself and lose a lot of blood? Will your blood pressure go down? Yeah, if you lose volume out of your circulatory system, pressure's going to go down. All right, so this is still related to water conservation, because your blood has water, right? Okay, so this is acting a little bit differently. Now here, if your blood pressure drops because of a loss of volume of water, what do you want to do in kidneys to correct for the drop in blood pressure or prevent it from going lower? With regard to water,
Drink water.
you want to retain it. You don't want to lose any more water. You need to keep it, because if you keep losing water, your blood pressure is going to drop even further. Okay, so this is what it responds to. This is trying to keep the blood pressure up. Now, real close to it's actually in the cells on the afferent arteriole, right next to the glomerulus, because that's the best place to test for blood pressure. Okay, of going into the glomerulus. There are special sensitive cells that's called the juxtaglomerular apparatus, and they can detect changes in blood pressure. So when they detect a certain threshold of a drop in the blood pressure, they release an enzyme. They produce this enzyme called renin, and it's dumped into the blood. Okay? Now, the target of renin is another blood protein that is a zymogen. Now, up to now, I've mentioned zymogens mostly in the context of enzymes, okay? Inactive enzyme that is activated by cleavage of a peptide bond. But zymogens can be other types of proteins, not necessarily enzymes. So this here really is not an enzyme. But this is produced by the liver, the blood protein angiotensinogen. Once again, notice here that ending, GEN, that's a common ending that you have on all types of zymogens. This is a protein, and that is the target of renin. Renin will cleave a peptide bond on this. It leads to a conformational change, and it produces a protein that's now called angiotensin 1. Now, there's other functions of angiotensin 1, but I'm not going to go into those because it's a very complicated system. Here, I'm only going to concentrate on the role that this plays with regard to blood pressure in the kidneys. This here is still flowing around, And there are other enzymes called angiotensin converting enzyme 1. Okay, should put it up there, -1, because there's also an angiotensin converting enzyme 2, which is similar but does different things. And it's that angiotensin converting enzyme 2 that is the target and doorway that the coronavirus, SARS coronavirus 2 uses to get into cells, not angiotensin 1. Okay, not angiotensin converting enzyme 1. So they're doing different things. Where is this found? It's tethered to the apical membranes of endothelial cells. Okay, endothelial cells. So they're in arteries, right? So as angiotensin 1 is captured by angiotensin converting enzyme, it is converted into angiotensin 2, and angiotensin 2 is the active hormone, okay? It's a hormone, and it has many targets. One of its targets, and the only cells in the body, and all this is in the blood, the only cells in the body that can respond to this has to have a protein receptor lock and key, or not, shape, counter shape interaction. They have to fit together. And if that receptor is there, then it's not a target cell. So one of these are the smooth muscles in the arterioles, and that leads to vasoconstriction. Now, how would that help blood pressure?
It will increase the blood pressure because it will constrict the.
Okay, it's cutting down on the volume of the piping system. If you cut down the volume, the diameter of the pipe, the pressure is going to go up. Okay, so this helps bring up the pressure by vasoconstriction, and this is all throughout the body. Okay, it's all throughout the body. Another target is the posterior pituitary, stimulating it to release ADH. And what does that do? What is the target of ADH? The collecting ducts. And what is the response?
Increasing in the blood volume.
Okay, increases, it leads to an increase in aquaporins so that water is reabsorbed. What's that do for blood pressure? Keeps it from falling any further, or it should help. You're trying to bring in more water, which would, another target is adrenal glands. Adrenal glands sit right on top of the kidneys. Now, when you dissected the frog, you saw that the adrenal gland, they called it adrenal body, was a rather diffuse orange-yellow line that went all the way down. But by the time you get to mammals, in mammals, it's a distinct cluster of cells that sit right on top of the kidney. So it's right on the anterior part there. And it releases a steroid hormone. This is steroid hormone, aldosterone. Okay, aldosterone. And the target of aldosterone is cells in the distal tubules. Okay, now this does two things. One, it stimulates transcription, translation of the gene for the sodium potassium ATPases to be inserted into the membrane. Now, what would, if you increase the number of sodium potassium ATPases that happen to be in the distal membrane, what is that going to do? What is the sodium potassium ATPases? What's their function? Active transport of what?
They remove the sodium.
There'd be a reabsorbent sodium, chloride follows, and what does that do to water that's in the filtrate of the distal tubule?
It'll bring in water.
It'll bring more water, okay? So more, it increases the reabsorption. Now, this aldosterone actually does two things. One, it increases the activity of the sodium potassium ATPases that are already there, but being a steroid, it binds to the regulatory DNA that controls the activity of genes that code for the sodium potassium ATPase, increasing transcription and translation. Now that takes a little bit of time, but still the aldosterone immediately increases the activity of the sodium potassium ATPases that are already there. That leads to more uptake of water. All this counters the drop of blood pressure, trying to improve it. Okay, so it's a complicated little system here, And this is just a figure that sort of shows you an outline here. I think this figure leaves out of the picture the aldosterone. It's only showing the renin and the angiotensin 1 from angiotensinogen and angiotensin converting enzyme, angiotensin target. Yeah, it does show an extra target here, the hypothalamus, which leads to thirst, so you try to seek out water and so forth. But this doesn't show, oh, it does too, here it is, aldosterone. All right? Okay. There's yet another regulatory feature here, and this is called a hormone atrial natriuretic factor, or ANF. So if you just use the abbreviations for these hormones, that's fine. ANF. And it's just the opposite of the RAS system, the renin, angiotensin, aldosterone system. It responds to high blood pressure. Okay, why would you have high blood pressure? Or what would be one factor? There's many factors for high blood pressure. But one of them, you have too much water. So if you have more water in your circulatory system than necessary, then it's going to increase the blood pressure. Okay, so just here it should have just the opposite effect. We're wanting to rid the body of water so that the kidneys gets rid of more water. Okay, and so this here is measured where the sensors are, is tissues in, I believe it's the right atrium. No, it might be, well, yeah. Anyway, I believe it's the right atrium. So the sensors are there. That's when blood is coming into the heart. And those are also the cells, the sensor cells, are also the ones that release ANF. That's released into the blood. And only the targets that, this is a protein, and only the targets that have the receptor. And so what this binds to are receptors in the juxtaglomerular apparatus, which then lowers the release of renin. So it just reverses the release of renin, which is shutting down the RAS system. Okay, at least lowering the RAS system's reaction, which is to retain water. Okay, and another target here is that it also suppresses release of aldosterone by the adrenal gland. This is also part of the RAS system. Okay, so it's hitting several different areas in the RAS system. But it also binds, another target is the cells in the distal tubule, which it slows the activity of the sodium potassium pHs. Okay, so less of it is reabsorbed. If less sodium is reabsorbed, what does that do to water balance? If there's less sodium reabsorbed in the distal tubule, what effect is that going to have on water content in the distal tubule?
Water will leave it.
Water will still stay in the filtrate. Water will not be reabsorbed. Water will go into the collecting ducts. And another target is the glomerulus. It is increasing the leakiness of the glomerulus. Now what's that going to do? What sense does that have? Why would that have any significance in having high blood pressure? Because what do you want to do with the water? You're wanting to get rid of it, right? You want to bring it down so that the volume of the blood is less. So if you're increasing the leakiness of the glomerulus, how's that contributing to that goal?
You lose water.
You're going to leave, yeah, you're getting more water into the filtrate that will stay in the system and eventually be urinated out. Okay, getting rid of the water. See, some of these hormones have a lot of targets. Okay, it also suppresses the release in the pituitary of ADH. And what would that do? If we're lowering ADH, what effect does that have on water retention in the kidneys?
It'll bring you more water.
It'll leave water in the collecting ducts. Because this here would lead to the removal of aquaporins, okay? And therefore, less water would be capable of leaving the collecting ducts. And it would end up in the urine, okay? All right, so those are some of the hormones that are taking care of, and this is complicated. Now, I'm going in, the next lecture is hormones. This here is the introduction to the hormones. And then after the hormones, I talk about reproduction. Now, these three lectures, the end of this one, and also reproduction, hormones are all connected together because reproduction is highly regulated by hormones. And so these are all fitting together. What I'd like you to know about the hormones, of course, is we're not going to cover every hormone in the body. There's a lot of it. Just going to hit the major ones, the major hormones. And in the process, I always want you to know, of course, the name of the hormone, its source, what produces it, okay, and what its target is, or targets. Some of these have more than one target, okay, like what I've just got over here. Angiotensin II has multiple targets. ANF has multiple targets, okay, and so forth. And there'll be others like that. And then the response of the target, response of each specific target. That's real important. Okay, any slide you want to see here before I switch to the next? Yes?
Question, which one releases the ANF where?
That's cells in the heart,
So the right atrium specifically?
the right atrium. Yeah, cells in the right atrium.
June 30.
Ready? Okay, anything else? Let me know when you have it. Okay. Okay, so now we have an introduction to hormones. Hormones are real important. They sort of work in conjunction